University of California, Riverside. I feel very lucky and happy to have this opportunity to introduce my research work to you guys in such a top conference. So today I'm going to introduce the NovoStitch, which is a genome assembly reconciliation algorithm using optical maps. So first of all, let's have a look at the background of this problem. Some studies show that there are 8.7 million eukaryotic species and about 200,000 genera on our planet. So among all of those species, only a few thousand eukaryotes have genome sequence available. So it means at least until now, the de novo genome assembly problem is still very important. We all know that in recent 10 years, the sequencing technology improved a lot, and the length of read is becoming longer and longer. However, despite this, the de novo genome assembly problem is still very challenging. That's because even for the third generation sequencing, whose read length is about 10,000 base pair, it's still much smaller than the genome size. So when we do the whole genome assembly, we have to assemble millions or maybe even billions of fragments into the whole genome based on their overlaps. So this process has a lot of challenges. We divide them into two categories. The first one is the technology-dependent challenges, which include non-uniform or high sequencing errors, chimeric reads, and uneven sequencing coverage. The second category is the genome-dependent challenges, such as repetitive content and heterozygosity. So because of those challenges, when doing the de novo genome assembly, usually people use different genome assemblers and different parameters to generate multiple assemblies and pick the best. But this idea sometimes doesn't work. That's because very often that there is no assembly that both maximize the contiguity and minimize the assembly errors. So in this kind of situation, people have to compromise one for the other. But the question is, why do we have to pick one assembly? Is it possible that we generate one assembly which takes advantage of all of the assemblies? So based on this idea, the assembly reconciliation is proposed. It is a way to obtain a higher quality consensus assembly by merging all the assemblies produced by different tools or parameters. So this figure shows the structure of this problem. The input is a list of assemblies produced by different tools and parameters, and the output is the merged consensus assembly. Some methods also use the reads as input. So the assembly reconciliation looks like a good idea, and there have been some tools for solving this problem. But the question is, how is the performance of those tools? To study this, some previous students in our group use benchmark datasets, GAGE, and some synthetic datasets to test those tools. This work is published in Genome Biology. We have no time today to look at the details of their experiments, but we could have a look at their conclusion. Unfortunately, they conclude that none of the tools they tested consistently improved the quality of the input GAGE and the synthetic assemblies. So in the aspect of contiguity, their experiments show a slight increase in contiguity in the consensus assembly when the original assemblies already have high quality. In the aspect of correctness, their experiments show that the number of missed assemblies ranged from the best of the input assembly to the worst of the input assembly. So we can see the problem of assembly reconciliation seems not easy. Is there any way to improve? Our solution is to use optical maps. The optical map is a kind of restriction map. When optical mapping, people cut the genome into fragments and use one specific type of restriction enzyme to nick the fragments. so that we could get the distance between the neighboring restriction sites. So the optical map is also fragmented, but the fragments could be like millions of base pairs. So usually the optical maps are used in the final steps of genome assembly pipeline to orient and anchor contexts with respect to each other and create scaffolds. So here we use the optical maps in a different way. Instead of doing scaffolding, we use them to drive the assembly reconciliation. So based on the optical maps, we got some high-level idea to solve the problem of assembly reconciliation. So first of all, we used, as we said, we use different genome assemblers and different parameters to generate multiple assemblies. So in this example, we use 3 genome assemblers for third generation sequencing, Genome, Aborigine, and Falcon to generate 8 assemblies. The second step is to detect and remove the chimera context. Let's use this alignment between optical map molecules and 2 contexts as an example. So in this example, we can see the 2 red arrows show 2 conflicts between the optical map molecule and the 2 contexts. So when we meet the conflicts, if we assume the optical map is always correct, we are going to cut the 2 contexts as these 2 positions. However, some study shows that the optical maps can also have misjoins. So to improve the accuracy of this chimeric removal process, we create another tool called the Chimera Cogniser. Chimera Cogniser could use the optical map molecules and the contexts to correct each other based on their abundance. This tool is not published yet, but it's already available in GitHub. The third step is to align the context to the optical map. The fourth step is to find the minimum tiling path, MTP. The MTP is defined to be the subset of contexts which cover the same genome region covered by all of the contexts. So in this example, we can see we have only 2 MTP contexts shown by the red box. The last step is to stitch the MTP context into one longer context. So the high-level idea looks very straightforward, but in practice, there are some factors which make the problem of stitching overlapping contexts very challenging. The first one is the imprecisions in optical maps. As we said, the optical map can also have misjoins. So when we use the optical map molecules as template to stitch the contexts, some mistake could be caused. The second one is the inaccurate alignment between contexts and optical maps. So the alignments, the coordinates of alignments shown by the optical map can be sometimes could be very inaccurate. The errors could be thousands of base pairs. The third one is that some contexts could be mapped to different regions of the optical map, so which can also cause some mistakes. So because of those challenges, we model this problem to be an optimization problem. So given multiple chimera-free assemblies represented by a set of contexts S, an optical map M, and a set of alignments A of S to M. The problem is to stitch overlapping contexts based on A and obtain a new set of longer contexts T, such that T covers the same portion of the genome covered by S. And the size of T is as small as possible, and the conflicts of T with respect to A are minimized. So this optimization problem has 2 difficulties. The first one is that we can say this is a multi-objective optimization, which is hard to solve. The second one is that the conflicts mentioned here is very hard to quantify. So because of the 2 difficulties, instead of solving this big optimization problem, we create an iterative algorithm called NovoStitch. So this algorithm is going to stop when the final assembly is not changed anymore. So in each iteration of NovoStitch, there are 3 phases. The phase 1 is data reduction, phase 2 is stitching, and the phase 3 is the post-stitching processing. Let's look at the details. The phase 1 has 4 steps. Step 1 is the pre-process. We map the context to optical maps by reference-aligner. Reference-aligner is alignment tool proposed by Bionano company. Then we remove the alignments with low confidence. The confidence is a kind of score generated by reference-aligner, which represent the reliability of each alignment. The step 2 is the conflict resolution. When 2 contexts are mapped to multiple optical molecules and their relative coordinates are not consistent, a conflict resolution is necessary. We formulate this problem as a weighted vertex cover problem on a hypergraph. In the hypergraph, the nodes are the alignments, and the hyperedges indicate the conflicts among 4 alignments. We solve this problem by integer program. As we know, the integer program is the NP-hard problem, so we relax the integer program to a linear program and gives the full approximation algorithm to solve it. The step 3 is the coordinate unification. Since the optical map is also fragmented, a coordinate unification step is required for contexts that map to multiple optical maps molecules. We first build an association graph in which the nodes are the optical map molecules. Edges connect molecules which have at least 1 context in common, and the edge width shows the reliability of edges. Then we try to get the minimum spanning forest of this association graph. For each minimum spanning tree, we make the most reliable molecule as the root node. Then we traverse each minimum spanning tree and unify the coordinates of all contexts based on the coordinate system of the root node. Step 4 is the MTP computation. To compute the MTP, we build an overlap graph based on the unified coordinates. In the overlap graph, the nodes are the contexts, and the edges indicate the overlaps between the nodes. In the ideal situation, each connected component of the overlap graph should be a DAB, with 1 source and 1 sink. However, in practice, it is possible to have more than 1 source and more than 1 sink. So because of this, we model this problem to be a combinatorial optimization problem called the smallest subDAC problem, in which for each connected component O of the overlap graph, we want to find the smallest subgraph P of O, such that any source sink pair ST, in which T is reachable from S in O, T is also reachable from S in P. We prove this optimization problem to be NP-hard by reducing it to set cover problem, then solve it by a greedy algorithm. Phase 2 is kind of simple, just stitch the overlapped MTP context. So to do this, we do the sequence alignments for overlapped MTP contexts by BLAST. To speed up, we use OpenMP to run BLAST on multiple cores. For each pair of overlapped contexts, we use the longest sequence alignment to stitch. The phase 3 is post-stitch processing, which is actually a stitching verification step. We map original context to stitch the context, and only keep the stitch when the most regions of stitched context are covered by alignments. Otherwise, we are going to cancel this stitch. Since the alignments are fragmented, we need to find the optimal set of mutually compatible alignments. We solve this by designing a dynamic programming algorithm. So we test our NovoStitch algorithm on the copy dataset. We use two types of restriction enzyme. One is BSPQ1. The other one is BSSSI. And we also use two sets of parameters, the loose one and the strict one. So we can see for all of the 4 experiments, the contiguity of our stitched assembly is much better than the best contiguity of the input assemblies. And in the aspect of correctness, our stitched context correctness is similar with the input assemblies, even a little bit better. So to conclude, the assembly reconciliation is appealing, but it seems a hard problem if based exclusively on the primary sequence. An optical map allows accurate assembly reconciliation of multiple genome assemblies. Experimental results show significant improvements in the contiguity of the assembly with similar levels of correctness. I would like to appreciate my PhD advisor, Stefano Lonardi, my lab mates, and our cooperators. I also would like to appreciate everyone here who listened to my presentation. Thank you very much.
So we have time for 1 or 2 questions. Shilpa, if you want to get set up. Yeah.
I didn't see in your algorithm where you used, where you took into account optical maps that might be chimeric.
You mean?
How do you identify potentially chimeric optical maps?
Oh, that's actually the part of removing chimeric contig is not the part of NovoStitch algorithm. It's actually the second work in that one is called Chimera Cogniser. We actually solve that based on the abundance of the contigs and the contigs and the optical map molecules.
Okay. Just by coverage of both to find kind of a voting.
For example, if a lot of contigs show that the optical map is wrong,
yeah,
then we're going to think the optical map is wrong. If a lot of optical map shows that contig is wrong, we're going to think that contig is wrong.
Hey, just a quick thing. I didn't get a good look at your results table. I was just wondering if you could go back.
You want to look at the results?
Yeah. Thank you.
So you show here quite an improvement in contiguity, but I was wondering if you did any assessment of the structural accuracy of the assembly that resulted.
Yeah, the last line, the last row of the table shows the correctness, the accuracy. The value is the larger the better. The larger means more accurate. So we can see our value is similar with the input, but it's a little bit better.
Great. Let's thank the speaker again. Thank you very much.
Thank you.